we have a bus if you want to give Cindy $5 and your name, excuse me, Dr. Hunter, and your name. She will reserve a seat for you. Otherwise, you can just show up at 8:30 in the Bachman turnaround and take your chances that there will be room on the bus. It's going to be awesome. Monk seals, albatross chicks, shearwaters, all sorts of good plants. Yes?
Do they expect big surf that day, or they don't know yet still?
I haven't checked the surf report, but it won't be impassable to get there, I don't think. Yes?
Where's the Bachman turnaround?
The Bachman turnaround is if you're coming up University Ave, the first thing you see on campus. There's a grassy field where people play frisbee sometimes, and a U-shaped pull-out. The buses stop there.
Okay.
It's in front of Bachman Hall. We gotta call it the Bachman turnaround.
Is that Sinclair Circle?
Yeah.
Yes, next to it.
Yeah.
Adjacent to Sinclair Circle. So the lower, let's see, lower Eva corner of campus is where we're gonna meet with the bus. 8:30. So I hope a lot of you can make it. Today's guest, I'm really happy to introduce, is Dr. Allison Sherwood. She's our phycologist, one of two in the botany department. She's also our department chair. She got her PhD at the University of Guelph in Canada, came here as a postdoc, and transitioned into a faculty position. She studies both marine and freshwater algae, which is really pretty special to dive into both of those realms, I think, and she's going to tell us a little bit about the diversity of these groups in Hawaii and some of her ongoing work.
All right, let's try this out. Thank you, Anthony. Dr. Amen. All right, I'm happy to be here today, talk to you all. And I realized when I was going through my notes for this class that I think I've been guest lecturing, except for 1 year, since 2003. It's starting to add up. I'm really happy, though, at this point to have some interesting research to tell you about that my lab's been working on. And so I'm going to try to string some of the different storylines together into a summary talk today that deals with the non-marine algae of the Hawaiian Islands. So how do we start to look at the patterns of non-marine algal diversity and gain some insights into where they might have come from, how they got here, and how they might be spreading around the islands. And so Dr. Amen mentioned that my lab sort of delves into these different ecosystems here in the Hawaiian Islands. And there are a series of different topics that we're looking at right now in the lab that include things like airborne algae. I have a postdoc who's working on really deep water seaweeds, Heather Spalding, so she studies the deepest of the deep. We're looking at these general algal inventories of the Hawaiian Islands, so what can we say about the overall patterns of the non-marine algae versus some of the seaweeds that we have here. I have some students who are working on subaerial biofilms, so these small crusty biofilms that grow especially on the sides of tree trunks and things like that on the windward sides of the islands. There's a really phenomenal algal diversity there and some interesting possibilities for how things got there directly from the ocean. I have one PhD student who's looking at algal sea slug symbioses, which is sort of not something my lab typically does, but it's really been an interesting project for her to undertake and use this as a mechanism for looking at algal diversity, the smallest forms that are consumed by these sea slugs. And then finally, algal habitat transition. So how do you get something from a marine habitat moving on to land, as an example? Okay, but today I'm going to focus really on the non-marine forms. Okay, so these are the freshwater algae, they're the terrestrial algae, essentially anything that is not living in the ocean. You've probably seen a slide like this or identical to this from many of your speakers. The idea that one of the reasons we're also excited to work here in Hawaii is that we're so extremely isolated. And out here, thousands of kilometers from continental land sources means that the flora and the fauna that we see here in the islands necessarily came by these waif individuals originally that colonized the islands. And this led to really unique floras and faunas that we have here. The events that probably led to this, long-distance dispersal events, are reputatively quite rare. And what that means is that there's been a lot of opportunity for unique pathways of evolution here in the islands. And I think that's one of the main reasons why a lot of us work here, and probably why you might be interested in the biota of the islands as well. There's so many unique species that we have only here in the Hawaiian Islands. And it's really pretty interesting to look at the associations between what you find here in the islands and where they might have come from. This is making a lot of noise. What do you
Is it 'cause you're
suggest?
holding it?
Leave it alone?
Yeah.
Okay. I'll do that. Okay. So one of the things that's pretty phenomenal about a lot of the lineages here in the islands is something called adaptive radiation, which you've probably, have you covered already, the idea of this in class? Great. Okay, so we have a number of lineages in plants, animals, microbes that have displayed this phenomenon of adaptive radiation, where you get these suites of species that are very closely related to each other, and they've evolved into these niches that we have available here in the islands. And there's a number of really classic examples of these that we find for certain major groups here, things like the honey creepers and silver swords. So they're well known for some of the plant and animal lineages, but we don't know very much at all about some of these smaller groups of organisms like the non-marine algae that people haven't done nearly as much work on. So one of the things I've been interested in and my lab's been looking at is whether or not we see these kinds of patterns as well when we start to get down to the microbial level. Okay, so in thinking about how things get around, how things might arrive to Hawaii and then disperse to the different islands, we could look at a series of different mechanisms, and they've been summarized in 1996 from the algal perspective by Christensen. So he outlined these 4 basic mechanisms for how algae get around. And the first one is active, and what that means is that some of these algae actually have some capability for mobility. So flagellated forms, or things like diatoms that can actually amoeboid movements sort of go from one water body to another. And so he posited that there is, probably some spread that occurs by active movement of these organisms. Although, granted, it's probably not on a very large scale. It would take a very long time for a small alga to actually get itself from one water body to another. So we're looking at over generational time. Water is an obvious one. A lot of these organisms are living in water or are very highly dependent on water for at least some part of their life history. And so they can use the movement of this water as another mechanism for spread. The third one is being transported by other organisms. And the classic example that we think of in phycology is things like birds' feet. If you imagine a water bird going and landing in, say for example, in Hawaii, a taro field. So it goes down, it lands, it's a water bird. Imagine what the bottom of its feet look like after it's walked around in that taro field for a while. There's all kinds of things stuck to that bird, on the feet, on the feathers, and then the bird gets up and flies and goes somewhere else. So on a daily basis, there is transport of these kinds of organisms from one water body to another. I've also included anthropogenic transport under this category, and that's because humans are playing a huge role in dispersal of organisms, small and large, definitely around the Hawaiian Islands and many places around the world as well. The last category there is air. So we're talking about wind movement here, and there is a small but growing body of literature that is examining these patterns of microbial transport with these wind patterns. And people have actually been studying this going back hundreds of years. Charles Darwin was actually capturing diatoms on the Beagle, and he sent them off to a German scientist to identify them. He was capturing them out in the middle of the Atlantic Ocean. And so people have been aware of the fact that microbes have been dispersed through the air for a very long time. Okay, so we can think about the size of these organisms as playing a role in how easily they disperse and what sorts of mechanisms are probably playing a major role in their dispersal. So I'm looking here at a size range going all the way from microscopic down to what would probably be the smallest alga you could imagine, all the way up to the macroscopic end on the right-hand side to the largest alga, non-marine alga that we're aware of, which is about 10 meters in length. That's a stream alga from Japan. Okay, so we're going from the very, very small all the way to the very, very large. What are the sorts of things that affect their dispersal? There are a couple of key theories that come into play when we can look at these different size ranges. So if we start off on the microbial end of things, over on the left-hand side, we're looking at the smallest of the small. We're looking at organisms that are under about 1 millimeter in size. These are things that you really need a microscope to see a lot of detail on. And their dispersal and their resulting biogeographical patterns fall under this idea of everything is everywhere, which goes back to the early 1900s. The prediction under that is that these are going to be organisms that are very, very easily dispersed. They're so small that it's going to be very straightforward for them to be dispersed by wind currents or things like that. And as a result, you would expect them to be pretty widespread, cosmopolitan, commonly encountered in a lot of different environments. So when people start to think about the species boundaries for these really small organisms, they often invoke these ideas of environmental selection being really important, that there's not really a limit to their dispersal. And so if you're seeing patterns of biogeography, species only being found in certain areas, it must mean that the environment is selecting for them. Once they arrive there, are the conditions suitable for them to thrive in that environment? If not, they're not there. If so, then they're there. Okay, so that's been historically sort of the idea of how microbes get around and what determines the species patterns that we see for them. But at the other end of the scale, when we start to look at the larger organisms, people have for a very long time been aware of pretty localized distributions. That is, you don't see really large animals all around the Earth. They are very localized in their distribution. This comes along with higher rates of endemism and people relying more on explanations of geographical barriers as limitations for their dispersal, as well as environmental conditions. But at least in the phycological literature, what I'm seeing is what I'll call an emerging middle ground between these two ideas. And that's because the algae, I think, cross between these two different size ranges. You can find both. The idea that neither ubiquity nor endemism can fully explain the distribution patterns. Okay, so there's something kind of in between or a mixture of these two ideas that can be used to explain some of the patterns we do see. I've got two examples up there to show you what I mean by that. The first one's over on the left-hand side. This is a paper that came out a few years ago by some colleagues in South Korea. And they were looking at this cyanophyte. This is a small alga. It's colonial, as you can see, and there's about, it's a very small size, 75 microns across for that alga. Okay, so you definitely need a microscope if you're going to see it. It's really widespread, though. This is a species of cyanophyte that's been reported in many places around the world. And so they did some molecular work where they started haplotyping them, get a sense of the different kinds of representation within this species. And what they found is that there were certain genotypes or haplotypes that were very localized, that they only found in certain areas. And then there were other ones that were, pretty closely related. They seemed to be the same species, but they were very widespread. So there was a combination of cosmopolitan haplotypes and those that were endemic to certain areas. Similar patterns came out in a study a couple years ago by some folks who were looking at diatoms, and that's over on the right-hand side. This is two species, Pinularia borealis and Hantia amphioxus. They are actually Antarctic forms, but these have also been reported in areas up through Africa and Europe. And they found similar ideas, that there are some strains or types of these that seem to be very localized and others that are very, very widespread. Down at the bottom there, I've got a note that dispersal processes of freshwater algae are poorly understood. And I'll come back to that as I talk through the rest of my class today, but it's something that I think needs a fair amount more attention by phycologists. We're often looking at specimens that we're bringing in from the field and just doing our best either with molecular characterization or working at the microscope. But quite often we're not looking at the entire life history and trying to figure out how all these different phases might come together. Which part of the life history is most important when we're starting to think about dispersal? Okay, so I'm going to back off a little bit and talk more broadly about non-marine algae and why we'd even be interested in thinking about them here in the Hawaiian Islands. I know it's probably not a topic you've spent a lot of time thinking about so far. That's okay, because there are specialists who like to go in and focus on these things. But they are important, and I think there's a few things you should know about them, how they fit into the larger ecosystems that we have here in the Hawaiian Islands. First of all, they're important because they're contributing to the base of the food web. Okay, so there are a lot of organisms that depend upon them as a food source. They are primary productivity, especially in stream systems here. There are some fish, for example, the endemic goby, Siceopterus stimsoni, that feed almost exclusively on these. These are small gobies, and they are rasping the rock surface in the stream, and their entire diet consists of these small microalgal communities that are grown up on the rock. Okay, so they're food for these organisms that we have here that are pretty special, like these endemic fishes. They're used by other organisms as well for structural materials. So over on the left-hand side, you can see a micrograph of an aquatic invertebrate case that is built out of freshwater algae. So it's taken these strands of freshwater red algae and wound them up and made them into this case, which is a home for the organism to live in. I mentioned already that I've been here for quite a while at this point, but one of the first things I did when I arrived was try to get a handle on what was known already about the non-marine algae here in the Hawaiian Islands. And so I went back through all the papers that I could find that had any reports whatsoever of non-marine algae. And it turned out the record went back pretty far, about 140 years into the literature. But what I could put together was a checklist that had a lot of species names on it, but there were mostly scattered reports. These are papers that were usually addressing other questions altogether, and they just sort of incidentally had listed some algae that they identified along the way. So there weren't that many studies that had really come in and focused on characterizing this flora, which I took as a sign that is something somebody needed to do. So we spent a lot of time doing this. It's something my lab's still interested in. We're always trying to figure out, is this a new species? Is this something we've seen before? Can we add it to the flora or not? We sort of keep a running tally of what we know about the flora here in the Hawaiian Islands. But one of the questions that came up a lot when I was starting this work was, well, how special is this flora anyway? We know that there's a lot of endemic species in some of the plant and animal lineages, but what about the algae? Are they worth worrying about because there are a lot of species that are only found here, nowhere else? Or are these really common things that you'll find anywhere? So just based on the literature, before I started doing a lot of lab work, I calculated about a 5% endemism rate for the non-marine algae. This is just based on whether people thought things looked sufficiently different to name it as a new species. And if they went ahead and did that, I counted it as new. If it was still not recorded anywhere else, it was included on the endemic list. So about 5% of the taxa landed on that list. I did another study where I kind of narrowed it down and focused on things just in streams, and that number dropped at that point, about 2.3%, looking at that subset, stream macroalgae. So in that paper, we concluded that the Hawaiian stream algal flora is what we'll call suspiciously cosmopolitan. And I say that because those rates that I just listed, 5% or 2.3%, these are very low compared to most of the other groups that you can think of that are common here in the islands. So for comparison, start off looking at other algal groups. The red algae, the marine red algae, about 20% of those species are endemic. Okay, so described from Hawaii, not known from anywhere else. It jumps up to 32% for marine invertebrates, 74% for ferns and lycophytes, about 79% at least for flowering plants, and about 94% for insects. Okay, so all those numbers are substantially higher than what we've seen so far for the non-marine algae. So why might that be? Are they really that cosmopolitan? Are these the same things that we're truly seeing everywhere else in the world? Or are we just not capturing that diversity somehow? Are we not fully understanding what we should call these things? This is a quote from Peter Tyler, from 1996, which I think sums up nicely what the problem probably is. Across the world, there's a prevailing view that freshwater algae are cosmopolitan. The notion has seldom been tested and is unlikely to be true in genetic terms. The degree of endemicity is probably masked by the force-fitting of European names. Okay, so he's saying two things there. First of all, that we are probably not looking at the right characters if we want to fully understand the diversity. That's because a lot of these organisms are small, and they don't have that many morphological characters. So if you're looking under the microscope, there's a limited number of things that you can come up with to separate them out into different species. But if you start applying molecular concepts to these and looking at the phylogenetic diversity within them, quite often you find that there's a lot more patterning than you can discern just based on morphology. The second thing he's saying is that we are using taxonomy that maybe doesn't apply. What we're using are the available floras. And so if you look at the literature on the shelf in my lab, a lot of it comes from Europe. That's because that's where the phycologists have been active. And so those are the folks who have written the books on how to identify species. And so you kind of have to use it to some degree, but at the same time, would you expect all these species in Europe to be the same ones you find in Hawaii? No, there's no reason to really think that. So if we're going to take a taxon and sort of force-fit it into a category in a European book, then yeah, you're going to miss the diversity that's here. Okay, so let's get into the data part. So a lot of the patterns I'll show you now came out of a freshwater algal biodiversity survey that my lab was working on up until just a couple of years ago. This was a big collaborative effort that came together with a whole bunch of phycologists who were really interested in different algal lineages. So we had a team that was comprised of my lab that focused on the green algae and the red algae, Rex Lau, Pat Kaselik, who focused on diatom taxonomy, Jeff Johansson, who works on cyanobacteria, and Gernot Presting, who was doing specimen databasing and building the bioinformatics portal for us. We went to all the islands and as many non-marine habitats as we could access over the course of this survey, collected algal samples from all of these, and then brought them back into the lab to characterize them. This is sort of a breakdown of the sites and the different habitats that we went to over the course of the survey. We had several thousand of these collecting sites that we went to, and they covered everything from high elevation bogs to agricultural ditches and flumes. There are just a few lakes that we were able to access. Many streams, most of the sites were stream sites. We looked at taro fields, and then we also looked at these more terrestrial types of environments, so not really aquatic at all. They could be roadsides, they could be damp walls and the sides of hiking trails, things like that, and looked at all the algal diversity we could assess. In terms of taxonomic coverage, you can see it on the left-hand side, it kind of breaks down with 50% of the things we identified being green algae, and then a smattering from all these other groups. Lots of diatoms, fair number of cyanobacteria, and then these other lineages, freshwater red algae, eulanoids, and some xanthophytes. So the main idea when we brought these collections into the lab is to separate them out so that part of the sample was going to microscopical characterization and the other part was going to molecular characterization. So from the molecular work, we generated about 900 sequences representing 8 different markers, and that really depended on which taxon we were looking at. The idea being that we'd have this marker here, the UPA marker, which seemed to amplify really well from all these different lineages that we were using. And we could use this as sort of a first pass of whether we're seeing trends that needed to be separated out in more detail in terms of the systematics. Do they all cluster together really well? If so, it's probably a well-described species. Are they breaking up into different groups? Then in that case, we probably need to look at them in more detail. We took all these data, as well as the microscopical data, the geographical data, and uploaded into the database that we built for the project, which is available if anybody's interested in looking at these project data. That's the website right there. It's the Hawaiian Freshwater Algal Database, and we use it quite a lot still because it has all the collecting information for where we found the different species. It's got all the photographs that we took at the microscope. It has the DNA sequences in it. And you can search it in ways that will give you, for example, all the algae you identified from a certain island or a certain region of the island, or by habitat. So it's been useful for us, and we're trying to look at the trends that came out of the survey. Okay, so there's 2 questions that I'm going to try to answer with the data that we collected on that survey. The first one is, what proportion of the flora is cosmopolitan versus endemic? Okay, so we've addressed those ideas already. Are these things that we have in Hawaii special? Are they endemic to the islands, or are they simply things that we find everywhere else in the world? So how applicable is the ubiquity hypothesis, or is everything everywhere? And secondly, we'll look at the dispersal of Hawaiian freshwater algae. So we'll start off with the first one. You don't really need to look at this in a lot of detail, except to see that what we've done is made a whole list of common taxa over on the left-hand side. And they're broken up by color. So there's a red group at the top, and there's a green group. There's sort of a cyan color. Those are the cyanobacteria. And then there's a group of chromophytes at the bottom. So we grouped all these different algal taxa that we had identified and tried to look at the geographical distribution of each one of these species. Okay, so we figured out which of the Hawaiian Islands we found them on. And then we went into the literature and looked at all these other regions of the world and tried to determine if they had been found in other parts of the world as not. And you can see immediately that what we're trying to address here is whether things are unique to Hawaii or whether they are well known in other parts of the world. And we summarized these. So we looked at all these taxa according to the different categories. How many other categories were they found in other regions of the world? And we broke them up like this. So the first category is more than 6 other regions outside of Hawaii. Okay, so those were things that we decided we would call cosmopolitan because they're found in almost all the regions of the world that we had looked at. The second category was 4 to 5 other regions. The third category was 1 to 3 other regions, and the fourth one was nothing outside of Hawaii. So by default, that means endemic. And here's how it broke out. So interestingly, I thought perhaps what we would see is a lot of cosmopolitan taxa, maybe just a couple endemics. But what we actually found is that there's a decent spread among these different categories. Okay, so we're looking at about 27% that are what we think are probably very cosmopolitan species, things that are found in lots of different regions of the world. 39% and 23% in those middle categories, somewhat more limited distribution, but still pretty widespread. And then about 11% that are truly endemic as far as we can tell. And so that 11%, 11.4% is still quite a bit higher than what we had originally thought, just based on the literature, right? We've started off at about 5%. It's up to 11.4% based on this work. So I think we're starting to see the patterns a little more clearly when we're looking at character sets that go beyond the microscope. So I'd like to show you just a few examples of these different categories. This is a freshwater red alga, Compsogons ceruleus, that fell into that category of cosmopolitan. And you know, the reasons why it's cosmopolitan are probably not natural. This is an aquarium weed, and it's very easily dispersed through the aquarium industry. It grows up in freshwater aquaria very commonly. I think they call it staghorn weed. And it's this bluish-gray, tufty stuff that grows on other aquatic plants. So it's very easily dispersed. What we found when we did some molecular comparisons is that there is essentially no diversity at all, at least based on the marker we were looking at. Across all the islands, we have Maui, Hawaii, Oahu, and Kauai represented in our analyses, and then you can see the labels for the other samples that other people have looked at as well. They're from a lot of different places around the world. So areas of the Caribbean, Florida, Australia, Louisiana, Germany, New Mexico, and they're all essentially identical. So this is something that's probably been spread through the aquarium industry, and that's why we're calling it cosmopolitan. At the other end of the spectrum, though, we have things that truly have never been found anywhere else in the world. This is an example of another freshwater red alga that we described based on a collection from the stream in the Alakai Swamp. And this was collected during the field botany class that we run in the botany department. This was the first time the class was run a few years ago. So I was over with the group for a couple of days. We hiked out through the Alakai Swamp and along the way across the stream. When we got into the stream, we found these large red algal plants that really quite surprised me, because we almost never find that form here in the Hawaiian Islands. There's usually a much smaller alternate life history that we find. So I was really shocked to see these really big gametophytes and really interested to see what it possibly could be, given how rare they seem to be in the islands. So that's the bag of algae that we took back to the lab. So we looked under the microscope. That's what it looks like under the microscope. These are the taxonomic characters that are relevant. This became a pretty straightforward project because there was a study that had been completed not that long before this that was monographical. I mean, it looked at every species in this genus. So all we had to do was essentially slot ours in there and see how it fell out. They even included molecular data in the monograph. So we generated the same sequences, markers for the sequences of the same markers, did the same analyses. You can see that star towards the bottom of that phylogenetic tree that shows where that species came out. It was quite different from anything else that had been looked at so far, and even based on morphology, we found some characters that were unique. So we described it as a new species, and so far it hasn't been reported anywhere else, so we'll call it endemic. Let's get back to the idea, though, of biogeographical distribution. So is everything everywhere, based on what we know from the survey? We decided to look at this, coming back to what I was talking about earlier. How big are these algae? So we looked at all these different taxa that we examined in detail, and I've got them sorted out by these categories of distribution, and these are marked by different colors, right? So endemic at the top is that light blue color, all the way down to green, which is cosmopolitan at the bottom. So we went to the literature and looked up the range of sizes that you might expect for each one of these taxa and plotted it on here to see whether there were really distinct breaks in the size relative to the category of how likely it would be that they would disperse easily. And we found that really, no, there isn't. These are all lining up pretty similarly to each other. All these 4 categories are right on top of each other. But if you start to think about how these algae are dispersing, it's probably not the entire thallus, not the entire alga that's being dispersed in a lot of cases. It's probably the propagule, or the dispersal unit, which is in many cases a much smaller fragment of the alga. So we did that same exercise. We repeated it, went back into the literature and determined, based on the kind of reproduction that each one of these algae typically undergoes, whether we're looking at fragments, whether we're looking at spores, zygotes, and then plotted those sizes out again. What you can see is that for all these categories, the likely dispersal unit is microbial. It's small. And what that means is that, at least in theory, these things could get around pretty easily. Okay, so what we can conclude, at least based on what we've seen so far, is that freshwater algae in Hawaii are categorized under a variety of distribution patterns. Okay, so they're falling out under these different categories. They're not all endemic, they're not all cosmopolitan. There's quite a spread between them, ranging from cosmopolitan to endemic, and endemism rate is now up quite a bit to about 11.4%. We also can see, though, just based on the last couple of slides, that these dispersal units are small. They're sufficiently small to be potentially widely dispersed. So why isn't everything widely dispersed? Well, it probably comes back to some of the ideas that we talked about earlier, that the distributions are influenced by other factors. And the thing that we're focusing on now has to do more, in my lab, has to do more with how things are actually getting around and what determines what gets where. These natural biogeographic patterns that we're searching for in a lot of cases are completely masked at this point, especially when we're talking about anthropogenic dispersal. There's just so much movement that I think is affected by humans every day that it's going to be almost impossible in the areas that are well populated to come up with natural biogeographic patterns anymore, probably for any group of organisms, including algae. So there's some photos down at the bottom that just give you a few ideas of how things get around. Hiking boots, you guys are going to Kaina Point. You'll be dispersing microbes along the way, almost certainly. Not much that can be done about it except do your best, clean off your boots in between different areas, especially going between different islands. But I'm guilty of this too. You go out on a less than ideal day where it's a little bit mucky, there's going to be a lot of transport of things. Agriculture, so wetland taro, for example, transport, these things are linked into the stream systems. And there's also an exchange of plants between different farms, things like that. Anytime there's a movement of plant material, there's going to be things that go along with it, including spores and fragments of algae and other things. And then the aquarium industry is over on the right-hand side. This is the plant we talked about earlier, Compsopogon. This is what it looks like in an aquarium. Okay, so it completely overgrows a lot of the aquatic macrophytes that people sell and keep in freshwater aquarium. Okay, so let's talk for just a few minutes about dispersal of Hawaiian freshwater algae, which is kind of where we're headed. I'm going to focus on these last 2 categories of dispersal. So organisms, anthropogenic, and air or wind patterns as modes of algal dispersal. Okay, so one of the things that my lab is really interested in is trying to get a pretty good handle on the things that are naturally present here in the Hawaiian Islands versus things that are probably being brought in sometimes on a regular basis. So this is what we're terming our urban algae project, and the kind of collecting that you do for this project is not that exotic or exciting. It usually involves just going to pretty heavily urbanized areas and seeing what we can find. Okay, so I have 2 students there in the upper left-hand corner that are sampling the bushes outside of Costco. And then we're looking at a sidewalk in a residential area. There's biofilm growing all over that, and these are probably algae that came out of the landscaping that's just above that area. Okay, so I realize I'm probably not going to be recruiting a bunch of students from the mainland to jump on board for this project, but we're thinking of this as a way of developing a checklist of things that probably don't really belong here. And we'd like to be able to subtract that from our other list of the entire flora that we're looking at to get a better idea of what are the things that are hopefully, possibly native here to the Hawaiian Islands. The other part of this project involves going to things like these big box stores because they are bringing things in all the time. And so we're looking, for example, if you go to the garden areas, they're often big, even aquatic planters or planters with plants in them If you look at the soil under those plants, there's often sort of this greenish fuzz, algal biofilm that's growing all over it. And so they're bringing things in from other areas, especially from the mainland. I'm trying to get an idea of what those things are. But the second part of this is the airborne algal work that my lab is doing now. So we're trying to sort of build on research that was started back in the 1960s, where people did some interesting studies characterizing things that they pulled out of the air here. And people have actually put forward the idea that Hawaii is ideal. I know that's going out on a limb, but Hawaii is ideal for the investigation of airborne algal diversity and distribution. This person, Schlichting, was very excited about Hawaii as a model location for the study of aerobiology. And he had some good points. So we're isolated out here. What that means is that we're not as easily impacted, for example, by continental dust. That will impact a lot of other areas where you might want to do this kind of research. We also have the trade winds, not this year, but most of the time when we're not in El Nino, we have trade winds reasonably predictably, and they are at least assumed to be free of biological particulates. And so we have this predictable weather pattern combined with being pretty isolated. It means it's one of the best places in the world for studying in isolation what these patterns might be. There's a little diagram down at the bottom there that I've redrawn from a paper, and this was work that was done in the late 1960s, published in 1971, and this is from a study by Malcolm Brown where he started looking at airborne algae here on Oahu. And it's a project that we essentially repeated in the lab. So I had a summer student a couple of years ago who took this on. And what we did was redo the same transect that Brown was studying back in the 1960s. And it's just the poly highway. So essentially what he did was drive a car over the poly highway with the windows down and his hand out the window and petri dishes. And so he would just sort of capture them. He'd swap out these plates once a mile. So he's getting mile by mile what are the different airborne particulates that he's capturing. He would grow them up for 6 to 8 weeks, see what came out on those plates, and then microscopically characterize them to see which kinds of algae typically grew. So we did the same thing. That's xylem, and we did this transect a dozen times or so over the course of that summer. And we used both marine and freshwater medium on the plates because we were sort of curious whether these things were truly all freshwater in origin or whether some of them even came out of the ocean. So we drove the transect at about 40 miles per hour, as much as you can on the poly, given the traffic lights and things like that. And then grew up the algae for about 8 weeks. So each of the colonies that grew up, we then divided into half. So half of them we used for microscopical examination, the other half we sequenced to see how that came out. So just based on the molecular data, this is how things broke out. The overall diagram is over there on the left-hand side. And what you can see is that 58%, almost 60% of the things that we identified were cyanobacteria. So there's a lot of these cyanobacteria, different kinds. They're really beautiful. I'll show you in just a moment. That are floating around in the air that apparently we're breathing all the time. I had no idea. We breathed in so much algae. Green algae, about 32%. We had some diatoms. We had some land plants. These are probably spores, pollen grains, things like that were floating around out there. And a few things that we couldn't even assign to a lineage. If you're interested in algal taxonomy, I've broken out the green algae and cyanos so you can see some of those categorical breakdowns. But probably the pictures are more interesting. These are the cyanobacteria, or at least some of the ones that we pulled out of the air after they've been cultured up. So the part that was blowing around in the air was probably much smaller than this, and this is just how they grew up in culture to see the full adult allis form that was there. And they ranged broadly. I mean, we had very small coccoid forms, lots of filamentous forms. Some of them were branched, and some of them were really quite strange looking indeed, like that Calothrix on that bottom left-hand side. Green algae and diatoms as well. Lots of different forms that came out in different lineages within the green algae. And 4 different species of diatoms, too, that we captured out of the air. Okay, so I mentioned earlier on that when Malcolm Brown was doing this, he changed out those plates every mile. Okay, so we did the same thing. We had 12 plates each time we did that transect for each of the 2 media. So we had freshwater plates and marine plates. And we wanted to use those to start to look at mile by mile if there's some patterns involved in the kinds of taxa that you pull out of the air, different locations along the poly. So this graph that I'm showing you at the top has mile 1 through 12 on it, and that corresponds to the illustration on the bottom. So starting on the Honolulu side, that's mile 1. The very peak is up between mile 6 and 7 at the top of the poly, and then Kailua Beach Park actually is mile 12. Okay, so you can see mile by mile some of the patterns that we uncovered. So this one is just looking at which things grew up on freshwater versus marine plates. And you can see that most of the things we pulled off freshwater plates, but there are some marine things that came out in most of the locations. The only site, though, where we found things that grew both on the freshwater and the marine plates was the very last site over at Kailua Beach Park. There were a few things there that grew on both. Okay, this is an illustration using the molecular patterns that we got out of these samples. So we just sequenced, in this case, one marker for all these different algae and then put it into this reference framework that we've been building over a number of years for all these different taxa. And we just used it to try to look at the phylogenetic diversity, which things popped out in which groups. Okay, so you can see that some of these little lines, these are taxonomic labels, are showing up in colored form. So the blue ones are freshwater, the red ones are marine, and then all the gray ones are really just the background. Those are the reference sequences that we use to categorize things. And I'll just pull out a couple of quick patterns for you. The first is that we had some taxa, as I mentioned earlier, that grew both on marine and freshwater, like this one particular group of Cyanobacteria. And then we had other things that were very specific in their requirements, like this Calothrix that only grew on freshwater. And we can use that same diagram and do the same kind of thing where we're looking at by mile. And this looks like, this is the rainbow slide, right? So what we've done is given a different color for each of the miles along that transect, and then labeled those sequences according to which mile they were recovered from. And there's not a whole lot that you can gather by looking at that whole thing all together, so let's pull out a few patterns. The first is that all the land plant diversity, so the spores that came from ferns and lycophytes, they came out of locations that seem to be near the summit of the poly highway. Okay, so those are the only places where we found those. The second is that the diatoms only came out of that final mile, Kailua Beach Park, which is interesting. I don't know if these are definitively marine taxa or freshwater, something we should probably look into a little bit more, but there's a possibility there that you're getting coastal sea spray off the windward side, and that might be what we're seeing there. And then thirdly, there were a couple of taxa that seem to be really broadly distributed. That's where you get the rainbow effect, right? Every mile along the transect, essentially, picking up that same alga. So this Cyanobacterium seems to be really widespread. Okay, so just wrapping up this part. This, I think, was our first application of molecular methods to characterizing airborne algal diversity in the Hawaiian Islands. And there were all kinds of different groups, as you saw already, that we've picked up using that method. But what's really interesting, though, is that the vast majority of the things that we identified from that airborne study were not things that we knew from the previous survey that we had done. So we thought we'd done a pretty good job already. We'd done that freshwater biodiversity survey, characterized all kinds of things, and then it turned out when you look in the air, these are all new things. 97% of them are OTUs that we didn't even know from before. So there's probably a lot more work to be done on both of these forms. Really quickly, I'll just mention that we're carrying on with this work. We have a study that we're wrapping up right now doing a windward, leeward Oahu comparison of airborne algae, where we collected samples over a period of about a month and a half, and starting to look overall if the diversity is different on the windward versus the leeward side. And then some of the studies that we have going on as well that are going to take a year or two to probably come to completion, looking at seasonal patterns of diversity. But thanks very much to Dr. Aman for introducing my lab to these next generation sequencing techniques, but we're starting to get to a point where we can look at the diversity on a much finer scale by using the next generation sequencing techniques that give you so much more sequence data than we were able to generate using the Sanger sequencing approach. So all of a sudden, by getting these millions of sequences instead of, say, a few dozen, you can start to fine-tune the patterns that you're looking at in a little more detail. Okay, I'll end up there. I've had a lot of support and a lot of help from some really great colleagues, so I'll leave that slide up there for a few minutes so you can see who they are. But thank you very much for your attention today, and I'd be happy to answer any questions again.